Okay, now we're going to talk about the cell envelope, which consists. So why do we need this cell wall? Well, because it's going to determine the shape of the bacterial cell. It's also going to provide structural support to keep the cell from taking on too much water and then bursting because of osmotic pressure. So in effect, it's a suit of armor or a corset to actually support the cytoplasmic membrane below and keep it from exploding. If you have the cell wall around you, you have wall pressure opposing osmotic pressure. And so therefore the cell does not lyse. You take away a cell wall, for example, if you grow cells in the presence of penicillin, they don't make a proper cell wall, they make a defective one. And so the net effect is that without that girdle around the cell swells up and then explodes. Now, peptidoglycan was called murine when I was in college, which meant wall material. And it is made up of peptide chains and sugar chains, so that's why they renamed it. And this substance only found in bacteria. It's found in most bacteria, but not in the mycoplasmas. They lack a cell wall. It has support, but it also gives flexibility to the cell. So if I looked at a bacterial cell, it's almost as if I have this lattice of peptidoglycan that is wrapped around that cell. And again, made out of alternating sugars. And these are sugars that are not widely used throughout nature. They are N-acetyl glutamic acid and N-acetyl-muramic acid. And they form dimers that are polymerized to make these long chains. Now, those long chains of sugars are held together by peptide bridges. And the peptide bridges are built out of tetrapeptides. That means 4 amino acids covalently bonded. And those 4 amino acids here are alternating D and L amino acids. Now, typically bacteria or living cells use L amino acids. That's the isomer that is preferred. But occasionally you'll have the D form amino acids incorporated into substances. This makes them a little tougher to digest. So here's my D-glutamate and my D-alanine showing up in that tetrapeptide. Now I have a bridge. Here's my cross bridge holding it together here. And my cross bridge, okay, is going to vary within the species, but it covalently joins together this tetrapeptide chain with this tetrapeptide chain. So in effect, you're sort of like stapling all these chains of sugars together because of these inner bridges. Now, if I interfered with the formation of that cross bridge, then I would have a defective cell wall, and that's what penicillin does. It interferes with an enzyme called transpeptidase, which forms that bridging structure between the two chains. Now, so if I look at the gram-positive cell wall, it's very, very thick. It's layers and layers and layers of this peptidoglycan here. And then running through that peptidoglycan lattice are chains of glycerol phosphate and ribitol phosphate that form a structure known as teichoic acid. Teikos also means wall, okay? And so this lipoteichoic acid binds to the lipids that are in the underlying cell membrane, attaching the cell wall to the cell membrane below. But the teichoic acids only extend through the peptidoglycan layer. Now, they have a charge on them because of all those phosphates, and this adds to the net negative charge of the bacterial cell. Gram-negative cell has a much thinner layer of peptidoglycan, and it overlies the cell membrane, and there's space between this cell membrane and the outer membrane, which characterizes the gram-negative bacterial cell. And that space is called the periplasmic space, and it contains a lot of digestive enzymes and transport proteins. So there's something that goes on in that space. It has value and function. Now, this outer membrane is a phospholipid bilayer, but the outside face has a weird polysaccharide. It's called lipopolysaccharide. It has three parts to it. It has a lipid A component, which is in the phospholipid membrane, bound to the other lipids there, and that's toxic. Lipid A is the toxic component of lipopolysaccharide. Then there's a core antigen, a chain of carbohydrates internal, and then at the end of that LPS, dangling out of the cell, are the O antigens. And this chain here, these are actually recognition molecules. So you can actually have differences in that chain that allows you to identify the different bacteria based on their surface antigens. They're called somatic antigens. Somatic means body. Okay, and those somatic antigens trigger the production of somatic antibodies against them that can be used for serotyping bacteria. So we can actually use antibodies to identify particular bacteria based on their carbohydrates that are on that lipopolysaccharide. Now, buried in that outer membrane are porine proteins. And these porine proteins are transport channels, and they regulate the passage of certain things into and out of the gram-negative cell. Some bacteria have very small porine proteins, and they restrict what can enter that cell. And a good example of bacterium that has small porins is the Pseudomonas, which resists many antibiotics and also is resistant to a lot of disinfectants, because if you can't enter the cell, you can't do your damage. Now, the outer membrane also is a barrier and protects the gram-negative bacteria against certain dyes, and also against bile, and also against complement proteins binding to it and initiating the destruction of the cell. So the gram-negative bacteria have some protection that the gram-positives lack. Now, if I'm gram-positive, but I have a waxy layer on top of my peptidoglycan layer, okay, I can be what they call an acid-fast bacillus. And on top of my peptidoglycan layer, I have this unusual waxy lipid called mycolic acid. It's called cord factor because the cells that are surrounded by this extension of their cell wall, okay, hate water because this is hydrophobic, so they clump together and they make these long serpentine cords, okay, that are characteristic of, for example, of TB. This mycolic acid is a very long chain fatty acid, very hydrophobic, and it actually has pathogenicity that damages the host cells. So it's, again, it causes problems. It also is a protection for that cell because with this waxy material surrounding the cell, it makes it very difficult for enzymes to digest that bacterium, and also difficult for certain disinfectants to do their job. So it also makes them grow very slowly because it also slows down transport of nutrients. So whenever you get like tuberculosis, it's not a fast disease. It's a very slowly progressing chronic infection. Now, because of that waxy cell wall, these bacteria can actually be eaten up by white blood cells, but resist the digestive enzymes that are released when they're inside of the white blood cells. And so therefore they survive phagocytosis. And they can actually survive and reproduce inside of these macrophages and actually travel through the body in these little macrophage taxi cabs, spreading to other parts of the body. So they're very resistant. And when you try to select and disinfectant to clean for areas where you might have TB patients, you have to consider their hydrophobic nature and find something that will work on them, that will be tuberculocidal, okay, that will kill the tubercle bacillus, okay. Now, in order to stain them, since if you're trying to use a stain that's ionic, it's not going to have any attraction for that waxy cell wall. And so therefore, they use acid-fast staining. Acid-fast staining relies on a stain that has fuchsin in it, which is a fuchsia color stain, but it is combined with phenol, which is a very hydrophobic antiseptic, okay? And so this can actually penetrate that waxy cell wall, and it drags with it the color. So that's my primary stain. I actually usually do a heat treatment to drive the stain into the waxy cell wall. And then when I decolorize, I use acid alcohol, which has a little tiny bit of hydrochloric acid added to the alcohol to decolorize the cells that have that waxy coating. Now, cells that don't have it will lose the color, and then you have to use a counter stain on it. Now, mycoplasmas, beginning of the word, sounds sort of like mycobacteria, but even the mycobacteria were misnamed. Myco refers to a fungus, and once upon a time, people thought that the mycobacteria, because they were thread-like, they thought that they were fungi. And therefore, they call them mycobacteria. Now, we know that they are prokaryotes, okay? They do not have a nucleus. And then they also use the same prefix in front of these. Now, these cells don't have a cell wall around them, so they have any shape that they want. They can be long filamentous strands. They can be coccoid in shape, okay? They have no cell wall to determine their shape. So they are pleomorphic. Because they don't have a cell wall, they have to consider, well, I better do something to my cell membrane to toughen it up a little bit. And so what they do is they use sterols. They take sterols from their host cells and they incorporate that into their membrane, which toughens it up so that they don't burst. Okay, so whenever you're growing mycoplasmas in culture, you have to remember to add cholesterol to the medium. Okay, they need that. Again, these are pretty small cells. Okay, because they have a very small chromosome because they don't need all those genes that make the cell wall. So this is a really teeny tiny cell. You don't usually use your light microscope to identify these cells. You can see the colonies when they're growing because it's a pileup, but you don't see the single cells because they're really below the resolution of the light microscope. Now, included in the mycoplasmas are Mycoplasma pneumoniae, which causes an atypical pneumonia, walking pneumonia. You don't treat it with penicillin because penicillin works on cell walls, and so this would be ineffective. Mycoplasma hominis, which is actually a sexually transmitted disease and can actually cause premature deliveries and cause stillbirths, okay? A ureaplasma ureolyticum also can cause these complications with pregnancy. And often, because they're very common, people don't appreciate their potential to do damage. Now, this cytoplasmic membrane is very, very thin. In fact, they never really knew what the structure of it was until they had really good electron microscopes, because it is so thin. It's approximately 7 nanometers thick. So they had a lot of theories as to how the cell membrane was structured when I went to college. And ultimately, they have a better idea now than they did earlier about. When I was in school, they taught us the sandwich model, that there was a layer of protein and then a middle layer of lipids and then another layer of protein, because they knew there were phospholipids present, they knew there were proteins present. Now we have the fluid mosaic model, where we have a phospholipid bilayer with proteins embedded in the bilayer that actually can drift horizontally in that bilayer. That's why they call it the fluid mosaic model. And that is the one that is supported by evidence today. Now, if I am a cell membrane in mycoplasma, I have cholesterol stabilizing my cell membrane. The ancient bacteria, okay, again, vary from the true bacteria because they have a different type of cell membrane. They have these branched hydrocarbons that are present there, rather than the fatty acids that we find in the bacterial cells as well as in the eukaryotic cells. So what happens at the cell membrane? Well, if you're a bacterial cell, the cell membrane is the only membrane that you have. And so therefore, it has to assume all the jobs that the membranes and the organelles do for the eukaryotic cells. So the electron transport chain is going to be embedded in the cell membrane. It's going to be responsible for making ATP. It's also going to be where you're going to embed your photosynthetic pigments, although sometimes these photosynthetic bacteria cheat a little and they sort of push some of the cell membrane inside of the cell, and they call those internalizations thylakoids. And so it increases the amount of space you have for your photosynthetic pigments, so therefore you can do more photosynthesis. They're going to be function in processing nutrients to allow them through that membrane and into the cell, and also making things that you're going to export from the cell. So the cell wall materials have to be made and processed and passed through that cell membrane in order to be assembled on the outside of the cell membrane. They also regulate what goes in and out of the cell. They are selective permeability barriers. They are going to be able to keep some things out and allow small uncharged molecules to pass through easily. Things that are charged would have difficulty going through the phospholipid membrane, so they have proteins that are carrier proteins and transport proteins embedded in that membrane, allowing these charged particles or these polar compounds to pass through to the inside of the cell. They also involve in secretion of enzymes that you're going to use outside of the cell, as well as toxins that some bacteria that make. Now, the lipopolysaccharide, again, consists of 3 parts. The outermost part is the O antigen. It's going to elicit the production of antibodies against it. It's also for recognition. Endotoxin is what we call the entire lipopolysaccharide, but the toxic part is actually the lipid A part, which is the most internal, and that will trigger fever and shock, and also trigger initially coagulation of the blood proteins, and then after we use up all the blood proteins, we're going to have hemorrhaging occurring. And there are lipoproteins there that anchor that outer membrane to the peptidoglycan layer below it. Now, porine proteins are going to be embedded there, again, allowing things to come in that couldn't normally pass across that phospholipid bilayer. Now, those porine proteins can vary in their size, and Pseudomonas, for example, has very small porine protein, and as a consequence, it actually inhibits the passage of certain antibiotics like penicillin from entering the cell, and also can keep out certain disinfectant molecules. So it's relatively resistant. So you have to consider, if you're trying to inactivate Pseudomonas, think about what you're dealing with and try to pick a disinfectant or an antibiotic that will be able to breach those barriers that it is established. Okay, this is my gram stain, people. It's a differential stain because when I'm done with it, I'm going to have a different color in some cells than in other cells. So I'm going to treat them all the same way, but at the end, one will be a different color than the other because it's built differently, has different chemicals in its cell wall. Now, I'm going to look here. My first step is my primary stain, and this is a basic stain. This is crystal violet, really intense stain. Positive charge on the colored part of the stain, the chromatophore. It binds to the negative parts of the cell. Ooh, what would that be? Oh, that's the teichoic acid that we find in peptidoglycan. And so it goes and gets into this mesh, which is much thicker in the gram positive than it is in the gram negative. The next step I do is my mordant. This is grams iodine. When grams iodine is added, it reacts with the crystal violet that's already inside of that mesh, and it causes a precipitate to form. It makes it insoluble. I have now trapped that complex inside of the peptidoglycan lattice. Okay, and over here, same thing. But, next step, now I'm going to use my decolorizer. And my decolorizer is alcohol or acetone or a combination of the two. Now, alcohol and acetone are lipid solvents. And what they're going to do is they're going to dissolve some of that lipid out of that outer membrane of the gram negatives. And so I'm washing away my outer membrane, and underneath it, I have that thin layer of peptidoglycan, not much good at holding the complex in place. So that color washes away. But over here in my gram positive, I don't have an outer membrane, and all I do with the acetone and the alcohol step is I actually dehydrate the cell, and that actually makes the mesh tighter. So I'm not letting go of the crystal violet iodine complex. It's locked into place here. Now, if I stopped at this step, I would have purple gram positives and I would have colorless gram negatives. But I like to know, what's the shape of these gram negative cells? So I use a counter stain, a lighter stain than the primary stain. So I'm using safranin. And when I stain my cells with safranin, the purple cells take the pink stain, but you can't see pink because that dark purple is overwhelming the pink stain. But when I stain the colorless gram negatives, they are now stained like this reddish pink color. Okay, so when I'm done, the cells that are positive are going to be purple. The cells that are reddish in color are going to be my gram negatives, and I now can differentiate bacteria based on their gram staining ability. Now, that outer membrane we said was a permeability barrier. It protects the gram negative bacteria against bile. It protects them against certain antibiotics. It also protects them against dyes that would inhibit the growth of gram positives. So it really is a big difference how your cell wall is constructed as to how you can react to challenges in your environment. Gram negatives have an advantage because of their outer membrane. Now, I also have in my cell wall of some bacteria proteins that are considered virulence factors. These proteins enable the bacterium to be more pathogenic, to be more capable of causing disease. Now, Streptococcus pyogenes, this means pus former, can have in its cell wall a protein called M protein, which is antiphagocytic. What that means is that a white blood cell will be attracted to where you have streptococci present. They are drawn to that area chemotactically. But when they get there, this M protein pushes them away, so they can't effectively phagocytose that bacterium. So what happens? More white blood cells come thinking that they need help. And so you get an army of white blood cells who really are not doing that much at controlling the infection, but they are causing an awful lot of pus. And that's why you get pus on your tonsils when you have strep throat. So there's like 80 different serotypes of M protein. What does that mean? Well, it means that if you create an antibody against one type of M protein, and then you get strep again that has a different M protein on its surface, you're not going to have antibody protection against that. In fact, sometimes it can be a bad thing, because sometimes making an antibody against M protein means that that antibody, which is it's actually reacting to a very small piece of that antigen. That piece of that antigen might be common to another thing that's found in the body. And in the case of rheumatic fever, people who have rheumatic heart disease, after they've had a strep throat infection and they've made antibodies, now those antibodies turn on them because there's a chemical similarity between that M protein and the heart valves. And so now you're starting to use your antibodies that you made, not to protect you, but actually to attack your heart valves. They cause a thickening of the valves. It'll lead to murmurs. It'll be causing backflow because your valves can't close completely. And now you're starting to overwork your heart, and your heart will enlarge over your lifetime, and it can be fatal. Okay? A lot of people who got strep throat infections before we had penicillin had rheumatic heart disease, and as they got older, their heart started to fail because it would enlarge trying to compensate for these defective valves, and then they had to have valve replacements. Okay? Now, Corynebacterium diphtheriae has a cell wall that actually has a waxy material on it similar to the TB bacterium. And that has toxicity to host cells. Okay? Now, and the mycolic acid from your mycobacteria, your TB organism, also can be damaging to human cells. Now, some of these cell wall antigens, again, because we recognize them as foreign, we can make antibodies against them. They may be protective antibodies, or they may be antibodies that turn out to be autoantibodies that start targeting our own tissue, and then we have autoimmune diseases as a consequence. Now, the lipopolysaccharide that we have in the gram-negative cell, again, endotoxin. We do not want these gram-negative cells to get access to the bloodstream, because if they do, they can activate fever production, they can activate the clotting proteins in the blood, and we can start forming blood clots, and then we can use up all our clotting factors, and we'll start hemorrhaging. And that's the scenario that occurs when somebody gets plague. Plague is caused by gram-negative bacteria in the same family as E. coli, okay? But it is really virulent. It has this big, thick capsule around it, and therefore it resists phagocytosis. It also gets into the bloodstream and triggers this sepsis and this septic shock and this hemorrhaging. Now, when this hemorrhaging occurs, it actually causes bleeding under the skin, and you develop these purple spots. That's called purpura. And if you survive that infection, those purple areas can actually become necrotic. And if you die, okay, they become necrotic, definitely. And now you have the Black Death. okay, because of the dead tissue that results. The Neisseria meningitis has a gram-negative cell wall. And because of this, it causes sepsis and septic shock and intravascular coagulation. So you get purpura if you have a bloodstream infection with the Neisseria meningitis. They call that meningococcemia because emia means in the bloodstream. And it is often fatal, and it progresses very rapidly. And if you survive, you may actually have dead tissue, and you may actually have autoamputations where your fingers and your toes develop gangrene and then they drop off, okay, because of it. So what do we have inside of the cell? We have a gel-like material that's called cytoplasm. It can be very, very liquid, or it could be more like a jello. It does a gel to solid conversion. And in this gelatinous material, we have embedded our structures like our inclusions and our nucleoid area where the bacterial chromosome is and the ribosomes. But we also have dissolved in that aqueous solution, we have proteins and we have sugars and amino acids and salts. And because they are on solution, it allows chemical reactions to occur easily. We also have cytoskeletal fibers in that cytoskeleton, in that cytoplasm. Now, the bacterial chromosome is a double-stranded loop of DNA that's very tightly condensed. So it fits into a very small region of the bacterial cell. It's called the nucleoid. Okay, it is organized around basic proteins, basic because they're positively charged, which means they attract the negatively charged DNA. And so this makes them pack more tightly, more efficiently. Now, besides having DNA in chromosomes, I might have extra DNA. And these loops called plasmids, they're double-stranded, okay? And I might have one, I might have many, I might have different kinds of plasmids in the same cell, and I might have none because they are not essential. But they are beneficial because they carry genes that allow that bacterium to make certain enzymes or certain toxins that it normally would not be able to make. And because of those plasmids, it gets a survival advantage. And so therefore, if I have a plasmid that gives me the ability to break down penicillin, because I now have an enzyme I can produce called penicillinase, well, then I have the advantage over my friends that don't have this enzyme. And therefore, they die, but I'm surviving, I reproduce, and I take over the population. And that's why bacterial resistance can develop so rapidly because of this plasmid exchange. Not only does that one cell pass those plasmids to its offspring, but it also has the potential to share it with a neighboring bacteria. And it does that by a process that's called bacterial conjugation, which is a form of asexual transfer of DNA. Okay, I'm not having sexual reproduction here. I'm just simply swapping genes with my neighbor. It's called horizontal transfer. And because of this ability for plasmids to be shared easily, and also that we have enzymes that can open up the DNA in the plasmid and then insert desired gene sequences in that plasmid and make that plasmid a vector of something that can carry these added genes by this process called gene splicing, I can now have the plasmid telling the ribosomes of the bacterial cell, make this protein. Well, I just spliced in the gene to make human insulin. So what does that bacterial cell make? Human insulin. It's just following the directions that are in the DNA that was put into that plasmid. So it's been very, very useful for genetic engineering and biotechnology to have the use of these plasmids. Now, ribosomes are protein factories, and when they are not working on a piece of messenger RNA, they lay around in the cytoplasm in subunits. Once messenger RNA arrives, it binds to the small subunit first, and then the large subunit fits on top of it, and now they become a functional ribosome. And the bacterial ribosomes are different from the eukaryotic ribosomes. The bacterial ribosomes are composed of a 50S subunit combined with a 30S subunit, and together they make a 70S ribosome. Now, that sounds like bad math, but they're actually measured in units called Svenberg units, which they determine by spinning the lyse cells in a centrifugal field so that the particles will go down, usually because of gravity. But besides mass factoring into how fast they pass through that field, okay, there's another factor. Air resistance, the shape of the cell. When you combine those two criteria, okay, you come up with the Svenberg unit. And that's why when you have the eukaryotic ribosome that's 80S, you have a 40S subunit combined with an 80S subunit, a 60S, and then that equals the 80S eukaryotic ribosome. Again, bad math, okay, because it's not really additive. It's not just based on mass. It's also based on the bulk of the particle. Now, because they're different, you can target bacterial ribosomes with antibiotics like erythromycin and tetracycline. Find out what your enemy has that you don't have, and you go after that as your drug target. Now, if I'm a bacterium and I'm living in an environment that has lots of nutrients, I plan for the future. I start stockpiling nutrients. So I store extra sugars as glycogen, and I store extra fatty acids as polyhydroxybutyrate. And these little droplets of lipids sometimes accumulate inside of the cell so much that when you do stain them with basic stains, there's areas of the cell that they're like beads, okay? They're uncolored. And the reason is they're blobs of lipids that have no affinity for the ionic stain, so they remain colorless. And so when you see these little blobs, you know that that cell had lots of nutrients and was thinking about the future. Now, some bacteria want to be at the top of the pond because that's where the sunlight is. So in order to stay at the top, they actually have little life preservers inside of their cells. They're called gas vesicles, and that allows them to float and get more sunlight than they normally would have if they didn't have that device. Some bacteria will stockpile inorganic materials, minerals. And so Corynebacterium diphtheriae, which we've already talked about as the agent that causes diphtheria, a respiratory tract infection that would actually cause a soft scab to form in the throat and would actually block the airway, and that child would suffocate because of it. Well, this was a killer before they had the DPT vaccine, okay? And so it made a toxin that damaged heart muscle and kidney tissue also. So they wanted to diagnose this. Well, what they did was they would take a throat culture from somebody that had a sore throat that could have had diphtheria, and they would stain it with methylene blue. And methylene blue, you would think, would stain the cells blue. But because methylene blue changes its color based on pH, because these phosphate granules were so negatively charged, when they stained red. So what you had were like these little red beads inside of the blue club-shaped bacteria. And then because they knew that that child had diphtheria, it warranted them being treated with an antitoxin. The antitoxin was made from horse serum, and because horse serum is foreign, you might have that child developing a reaction to the horse serum. It was called serum sickness about a week after they got the antitoxin. So you didn't want to give it casually to somebody that didn't have it this disease, but you wanted to confirm the patient did have the disease before you treated them, because it could save their life, but the serum sickness was something that you didn't want to cause unless it was absolutely essential. Now, magnetosomes are stockpiles of iron oxide that respond to the magnetic pull of the earth, and because of that, they actually drag the bacteria down in the water, because at the bottom of the pond are where all your decaying material is, and so that's where the decomposers want to hang out. So that oriented them